Okay, let's go back to molecular fossils. Our mascot for today is the cholesterol molecule right up here. And you hear about these things in diet, like don't eat high cholesterol foods. That's because our body produces a lot of cholesterol. And when we transport cholesterol around the body, the little transport vesicles tend to crash. And they just turn into these greasy smears that are impossible to get out. And we eventually end up clogging our arteries with these, which sets off inflammatory type responses, and it just makes everything worse, honestly. So it's not that the molecule is bad, it's that we're very clumsy when we are handling the molecule biologically. Yeah. If there's a direction that I hope evolution takes us through natural selection, it would be in making us a little bit better at cholesterol transport. Yeah, but cholesterol is super important to eukaryotes in general. You will find cholesterol of different types. So we'll call all of these things sterols. And cholesterol is going to be the one that you find in animals. And so here's a picture of cholesterol. And there's going to be an ergosterol, which is the type that you would find in fungus. And there's stigmasterol, which is one of the types that you find in plants. Plants are complex, they don't all do exactly the same things. So, these things, fortunately, actually make pretty good molecular fossils, or they're still recognizable in at least some formations. And so you can take a bunch of rock and you can grind it and you can run chemical extractions on it and you can get out the little bits of altered sterols and all their, byproducts, breakdown products. Yeah, actually tell what's going on. If you want to look at like why might this be pretty good for fossilization, let's zoom in here. Do you remember the really reactive atoms from our other molecular fossil stuff? Yeah, with the proteins. Yeah, it was nitrogen, right? That was super reactive. No nitrogens in this molecule. Little less reactive, you got things like sulfur. There's no sulfur in here. Less reactive, but still kind of reactive. Oxygens. No oxygens? One oxygen. And the oxygens are like a little decoration. They come and go or move around, I think. Yeah. Yeah, mostly this thing is a carbohydrate, and carbohydrates are pretty non-reactive, at least diagenetically, and so that's why these are actually pretty good molecular fossil markers. That's the idea here. So the idea is if you go back and you find rocks of a particular age and you want to know when did the first thing with fungus-like cholesterol metabolism develop, then you can go and answer that question. Or plant type, cholesterol. Yeah, here you go. You can answer the question. This is like what they did with the Dickinsonia, where they figured out that it had an animal type cholesterol, although a relatively primitive one by the look of it, and was therefore at least on the way to being an animal, or was more animal than it is fungus or plant. Anyway, yeah. Here is the biosynthetic pathway for cholesterol, and a lot of these steps are conserved. There are many of these steps that involve proteins that actually are derived from each other. So a lot of these different protein names that are sitting here next to the sides of some of these steps, you see how they look the same. They've got this long sort of helix on this side and a bunch of vertical alpha helices here, and then like a little lid. This one has a little beta strand over the lid, but yeah, it's a mostly alpha helical protein. And this is I don't know, it may be descended from two copies of this one essentially, but we've sort of shortened it and doubled it up. These are some of the enzymes that carry out the steps of cholesterol synthesis, and many of these enzymes will carry out multiple steps of the biosynthetic pathway. And so we're going to go from acetyl-CoA, little tiny two carbon, yeah, I think so, acetyl-CoA, and we're going to stack those things together, and we're going to make mevalonic acid. We're going to stack all these mevalonic acids together, and we're going to make a squalene. Squalene is a common step in all of these biosynthetic pathways. And then if you want to go toward cholesterol, you'll go down here. And if you want to go toward like an ergosterol or some of these other types, you will branch off at different points, but the early steps will be the same. So the idea is that these enzymes evolved gradually, mostly by duplication from the look of it, and that the early steps of the process are conserved, and that the real difference is in what the last one or two steps of the biosynthetic process are in a particular organism. That's what makes ergosterol different from cholesterol. And it's a giant difference. We target fungi, all our antifungals, or most of our best ones, target ergosterol synthesis directly. Because if you knock that out, the fungus just dies. Yeah. So cholesterol, yeah. Important friend, yeah. As much as foe, yeah. I don't know. It can be our frenemy. Why not? So this is from a super cool study. It is right down here. It was in Nature in 2023, super recent. And they actually went to rocks of different ages. And they found ones that did good job preserving things. They got reasonable dates for these rocks. And then they examined the presence of cholesterol or sterols in general in there. And what they found were that you get everywhere that's green, I believe those are the plant sterols. Everywhere that's red, I believe, is the animal sterols. And so let's orient ourselves in time. Here's our Tonian, Cryogenian, Ediacaran. So here we are right before the Cambrian. This is the latest pre-Cambrian. And then this is going to be the Paleozoic, Mesozoic, and Cenozoic eras. And so you can see things have pretty much stabilized. Maybe the plant types were a little larger part in some of the early days, but yeah, it really hasn't changed that much. What we see are in the earliest times, we get this giant spike here where it's almost all red. And so that is the rise of animals. And at this point, we think that most of the, this is after the Great Oxygenation Event, which we'll cover elsewhere. But this, so there are still things filling the plant niche, but it's going to be cyanobacteria. And prokaryotes, as far as I'm aware, don't have at least complete versions of this biosynthetic pathway. There may be some archaea that have some parts of it. That would not surprise me, but we'll have to go look that up elsewhere. And so, yeah, this animal type cholesterol gets really big around the time when we think sponges start to exist. Around, which is probably Tonian, you get your sponge relatives. You should get early Cnidarians either in the Ediacaran or maybe Cryogenian. And you're getting, of course, all those weird Ediacaran animals right up here. But actually, looking in the Ediacaran, there's evidence for proper eukaryotic plant type, so presumably like macroalgae, like eukaryotic algae, cholesterol in addition to these other types. But then look at the purple. And look at all the purple you get back here in the Neoproterozoic and the Mesoproterozoic. These are protosterols. Protosterols are going to be these chemicals that are the earlier building blocks that then get turned into other kinds of sterols. And so you can actually see the biosynthetic pathway evolving kind of in real time. Because you got the early steps, you're making your squalenes, putting your little mevalonic acids together. And then you're going to be, joining these up and making a nice four-ring structure that's the proper sterol. And then you're going to be modifying some of the side chains on the sterols. And gradually, over time, you get all these extinct organisms now because their sterols disappear from the fossil record once we get into the Phanerozoic era. But these things existed and they had all of the intermediate steps in the cholesterol pathway. And I think stuff like this is important for understanding evolution, that it's not happening just in one go. How do you end up with a pathway with this many steps, this many enzymes, just out of nowhere? You can actually see it being built up in the fossil record. And it's not the animal body fossil record, because you look at an animal fossil, how are you going to tell, what kind of cholesterol did you have? You going to ask it? No. Yeah, crazy. But you can see in the molecular fossils how these things developed. And so the creatures that were around then, they look kind of like this. Little spiky balls, little sacs, little presumably eukaryotic things of some sort, or prokaryotes, maybe archaea, that were on their way toward becoming eukaryotes, because eukaryotes didn't come out of nowhere. It's probably a couple of different, like a prokaryote inside of a prokaryote is the idea, or multiple prokaryotes inside of a prokaryote over time. Yeah. Here is a look at basically the pathways as developed there. And so they're showing that in the Neoproterozoic, that's where we got the pathway as far as cholesterol and ergosterol and stigmasterol. And through fossilization, through diagenesis, the biological sterols turn into fossil sterols, and so you get removal of the oxygens, for example. But it leaves enough of the carbon skeleton that you can still mass this thing and identify what it is, and you got the branch pattern of the different branches there. And so it's still, very recognizable. So here are the biological sterols. This is when they appear. These are their fossil equivalents. These are some of the biological sterols that would be produced on the way toward making the sort of end product sterols. And you find evidence of some of these, 24-dimethyl being something like this. So these are the sterols, like the just like one step before the sterols. And then here are the protosteroids down here, which are the, here's squalene, epoxide, just sort of unjoined up squalene, which, then these are some of the early derivatives, lanosterol, for example. And so here are the fossil molecules that were found, which are not quite identical, but which appear to be from this category, like this sort of stage of cholesterol evolution. So yeah, cholesterol evolved. Isn't that neat? I think that's just the coolest thing. And so, it took a couple billion years, but we did it. We got there to cholesterol. And so here is sort of relative total steranes, so including the ones that you would find in bacteria, so not just the eukaryotic ones. And so the eukaryotic ones are going to be in purple and red and blue and green and a little bit of pink. And the bacterial ones are going to be in yellow. And so you can see it was more of a prokaryotic dominated world. And again, the organisms that had these protosteroids, you can find things that look like they might be eukaryotic cells back this far, but the molecular fossils are the best evidence, and some of these things may not have yet been actual eukaryotes. Yeah, which is, again, super cool to actually see this in the fossil record. So, super paper, if they'd have come to me, I would have said, put that thing in nature. That's where it belongs. And it got there anyway, so good job, everybody. All right, hope that was interesting. Thanks a lot.